Okay. Good morning, everyone, and a very warm welcome to today's Co-Pilot and IBioIC joint session. It's really lovely to have you with us. I'm Annelie du Plessis, a Senior Impact Manager at the Industrial Biotechnology Innovation Center, and I will be your mediator for the next hour. This session is part of the wider Co-Pilot initiative, which brings together pilot and demo scale infrastructures across Europe to help innovators move faster and more confidently through the scale-up journey. We are pleased to host today's event and to showcase some of Scotland's capabilities that sit within the European landscape. Right, to give you a sense of where we're heading in the next hour, here is our agenda. We begin with Yang Zhu, who will introduce the pilots for you and Co-Pilot platform and how it supports innovation across Europe. Neil Rennos will then give an overview of IBioIC and the FlexBIO scale-up facility, including some of the tools and programs that we run. After that, we'll explore real-world case studies from FlexBIO with Sheena Fraser. We'll then shift to the Edinburgh Genome Foundry, where Rennos Fragkoudis will share how genetic design is enabling the bioeconomy. And we round off with a European perspective from Mark Bustard, who will introduce the European Circular Innovation Initiative, Valley Initiative, which is all about connecting regions across Europe so they can share expertise, support each other, and speed up circular innovation at scale. We'll finish with questions and a short closeout from me. But in terms of asking questions to our speakers, please can you write them in the Q&A box during the session, and we'll try and answer as many as we can at the end. Please also note down the speaker you are asking your question to, which will help me to direct it to the right person. And now I'm going to hand over to Yang Zhu, who is the project manager at the Biobase Europe power plant. Yang, over to you.
Great. Thanks a lot. So Pilots for You, powered by Co-Pilots, is a very unique platform for bio-innovators to connect, collaborate, and innovate together with our pilot and demo infrastructures. So over the next few minutes, I will give you a very short introduction about Co-Pilot's project, the Pilots for You, the platform, and also walk you through some main activities and events we have planned and explain how you can benefit from joining our community. Next slide, please. So first, I'm guessing this picture rings a bell for most of you. So this is the classic scale-up curve, so the valley of death. So the blue line shows the risk, so it stays quite high in the lab and only drops once we reach the pilot demo phase. So the yellow dashed line is the cash curve. So the cost starts quite low, then dramatically increase when the risk is still very high. So that's a mismatch. So the high risk plus the high cost is why so many great ideas stall here. And it is why shared pilot and demo infrastructures matter. So they spread the cost and share the know-how to help bio-innovators across this valley. Next, please. So here is where Pilots for You fits in. So on the left, you have got the TRL, the technology readiness level 1 to 4. So that is the discovery phase, so proof of concept in the lab. But once you reach to a TRL 4, so you need kilos instead of milligrams, you need real bioreactors instead of flasks, and of course you also need engineers. So that's a green band, the TRL 4 to 7 is exactly what Pilots for You covers. So it is the bridge between the lab and the first commercial plants. Next slide, please. Who is actually in this TRL 4 to 7 bridge? Well, across Europe, we now have more than 120 open access pilot and demo infrastructures, includes IBioIC. So in this map, you can see the darker blue, the more pilot plants each country hosts, because all these pilot facilities are listed in one searchable database, which is the Pilots for You. So you can easily find the facilities you need, be it, let's say, bioreactors in Europe, in Belgium, or biochar production, for example, in Austria. So without hunting for a month. Next slide, please. So we know where the facilities are. The next question is, what can they actually do for you? The Pilots for You covers 12 technology areas with more than 50 specific technologies. So in other words, almost any step in a modern bio-based process is already running somewhere in our network. And next slide, please. So we know that in Europe, our pilot facilities have enough capacity with goods availability. But I have to say, very often, the best pilot facility for a bio-innovator might be located in another country. So Pilots for You organize many cross-border events. So from May 2025 to May 2026, 15 pilot facilities in 14 countries open their doors to welcome bio-innovators. So this open-day visits will not only showcase their facilities, but also provide insights and training to help bio-innovators to scale up their innovation. And we also provide the travel vouchers to bio-innovators to help cover their participation in our events. So for more information about the travel voucher, about the facilities we'll organize open day visits, please just scan this QR code for more information. Next slide, please. Alongside our in-person events, Co-Pilots is also hosting a year-long series of webinars together with our pilot plans, just like today. So from May 2025 to May 2026. So you can see these webinars cover quite broad range of topics, including very specific questions you may face during upscaling, or how to give a good pitch in front of investors, or how to protect your IP, or how to even prepare documents if your innovation is about launching novel foods in Europe. And all these webinars are recorded, and you can watch them on the new Pilots for You platform. Next slide, please. And now let me introduce the brand new Pilots for You powered by Co-Pilot platform, which has already been online since last month. So instead of, let's say, hunting 10 different websites, you now have one platform for facilities and also for the events. So you can easily identify the best pilot facility for you, either by using the location or use the filters, as I mentioned, their technology areas. Of course, if you need specific special equipment to do upskilling work and you cannot find the information here yet, so please let us know. So we can help you find the ideal ones within our network. And of course, we also provide, you can see the event calendar agenda. So you will see all the events linked to upscaling, the trainings, as I mentioned. So including all the agenda and also registration. So to help you find the ideal events you want to join. And the last slide, I encourage you to follow our co-pilots, Pilots for You, the LinkedIn page. So by following us, by following our page, you will receive the updates on upcoming events, webinars, and keep track of all the exciting things happening within the Pilots for You powered by Co-Pilots network. And I'm looking forward to working with all of you and seeing your innovative ideas take shape through our initiatives. Thanks a lot.
Thank you, Jan. We are now going to hand over to Neil Rennos, who is the Director of Bioprocess Scale-Up at the FlexBIO Scale-Up Centre in Edinburgh.
Hello, everyone, and welcome. My role is overseeing our FlexBIO scale-up facility, but we work very closely with both our host university, Heriot-Watt University, but essentially we're managed by the IBioIC, and so we're an integral part of the services we offer. And I just wanted to give you a bit more of an overview of the actual innovation centre. So established around 10 years ago, essentially IBioIC is an innovation centre sort of specifically formed to help drive the commercialisation of innovation through industrial biotechnology. We're also primarily a networking organisation, so we help bring together industry, academia, and government all together. In total, we have around 400 industry members, as well as a huge network of researchers, which really helps us network all of those different components together. Our industry members really do range quite widely from sort of spin-out companies all the way through to large multinational companies. And we were predominantly developed and set up to sort of drive innovation within Scotland. But I think it's fair to say we have a large reach across the UK and into Europe as well. And everyone is free to join us. And Annalie will talk you through about how you can do that after this section. So we can describe our innovation support across sort of 3 pillars. And so we sort of drive and support innovation through showcasing. So, for example, through like our network with events and social media and PR. We can help translate, particularly the challenge is always translating research into commercial ideas and feasibility. So we can support that through our things like our feasibility funding, which is currently open at the moment, as well as providing more general support with collaborative bids as well. And something quite innovative that we have that I'll talk a little bit about is our bioresource mapping tool, which is specifically aimed at Scotland. In terms of accelerating bio-based processes, we have our scale-up facility FlexBIO, which I'll talk on just now. And we'll also cover as well what we call our train to scale bio-innovative support program. So it allows us to help and give support to growing innovative companies. So we managed to do that support through having an impact team, which is led by Liz Fletcher, and the senior impact managers who were joined by Annalie as one of those on the call today. Essentially, they've been assigned a certain academic institutions, so to directly engage with those universities across Scotland. But they also have specific technical areas across industrial biotechnologies that they're assigned to. So, for example, from marine, agri-tech, sustainable chemicals, textiles, we basically have someone that's the point of contact to help engage with both universities and also within industry. And more recently, we've developed a program called what we call the IB Champions, which are essentially technical industrial biotechnology people within all of our universities. So essentially are the go-to person to help connect you more deeply into areas of industrial biotechnology that might be a focus within those universities. So we can help match academics to industry to help fund that collaborative research. And as I mentioned, we have this Biotech Innovators Train to Scale program. I think we can all appreciate a lot of early stage companies need a lot of support, but a lot of that support is perhaps more general innovation support. And what we recognized was the need for real bio-based and biotech innovative support. So our innovative Biotech Innovators program essentially focuses on different areas where IB's needed. So, for example, the fundamentals of scaling up, LCA, TEA, mass balance, supply chain and regulations. Essentially, if you're a newly formed company or you're looking to establish a company in the next year, although it's technically closed, we're happy to accept applications for any bio-innovators that are interested in joining the program up until the 11th of December. And Kirsty Nielsen on the right in this picture is our lead on that, and you can contact her directly or through any of the emails that you'll see in this webinar. Also, as well, we have the Scottish Bioresource Mapping Tool. This was developed collaboratively with Scottish Enterprise and Zero Waste Scotland, and sort of appreciates the need that a lot of industries within biotech have a lot of waste feedstocks, co-products, and valorizable waste. And essentially knowing what they are and where they are is a really big challenge. So we work collaboratively to develop a mapping tool for that. This is currently being updated at the moment, but again, more information on that on the email supplied. Now, onto the scale-up facility, which is what I manage, and Sheena will be joining us today on the call, who leads our upstream team. Essentially, we were developed to help essentially small-scale ideas scale up into commercial reality, initially into sort of semi-pilot scale and more recently into pilot scale. We're one of the sort of smaller facilities, and particularly where we focus is on the earlier technology readiness levels, but we really have quite a broad focus from anywhere from healthcare applications to waste valorization. And perhaps critically and also uniquely around some of the pilot facilities, we've recognized the need that companies, academics, universities need skilled people in bioprocessing technique. So we're also part of a program called Resilience, which is developing skills, hands-on skills within the medicines manufacturing sector, but also more broadly into the bio-based manufacturing. And in terms of where we work, I'm sure a lot of you are familiar with the technology readiness level. But as I mentioned, we are one of the smaller facilities, particularly around, we're focusing more on TRL 3 to 6, and with our largest system up to 300 litres with comparative downstream processing capabilities for that. But where we really excel is around taking what we call coffee cup scale processes, both in microbial and mammalian scale technologies, and scaling them into our larger systems, and really understanding and being robust with how we transfer those technologies from what's been developed in a lab for 2 or 3 years. It often isn't trivial to transfer that to another facility and then move forward with scale up as well. How we engage is, we often say we try to engage as early as possible, particularly in a 3-year project. It isn't about coming to us in the last 3 months. We often try to engage at the start of the project so we can help perhaps inform some of the decisions made during the innovative journey. We were designed to be open access and essentially direct use, but feasibility-wise, this often doesn't really work in terms of having very high-value, complex equipment to be used, but we have some unit operations that can be used directly. More readily, we support more directly with things like small projects. We call them short duration, so things between sort of 1 day, 5 days, or multiple days over several weeks. We can have people on site as well that can observe and perhaps assist with that as well. And we also have our larger projects, which we call research services. So that can be anything from several weeks to several months, and over the whole project up to several years. Ultimately, though, until we go into some of the more exciting case study that Sheena is going to be taking you through, we've delivered over the last several years close to 80 projects, and that's with close to 50 organizations. So we've had several people that have come to us, perhaps with early stage funding, done some work, gone ahead and got more funding, and then they've gone and done more scale up work with us, and then gone on to things like Series A and Series B funding as well. Although it's not something we do easily, just because of space constraints, we have incubated 3 companies as well, and all of them have gone on to serious your Series A and B funding as well. But also within our training portfolio as well, which we often try to mix and blend with doing scale-up projects as well as training. We've trained anything from undergraduate students through to industry professionals, totaling over 3,000 hours of training over the last few years. But ultimately, we're publicly funded, we're here to help, and we like to think we know someone that can help you if we can't help you, and we are ultimately designed to be cost neutral. So what we charge, often scale up projects are more expensive than people envisage, but we try to charge what it costs us to do, to try and be as cost neutral as possible. And finally, I just wanted to showcase some of our training courses that we do. And we find we have attendees from across Europe for these training courses. We also do further hands-on training for undergraduate and master's courses, but these are really our showcase courses for sort of 3 to 5 days to really get you hands-on with pilot scale equipment across upstream, downstream equipment, and also into mammalian cell culture as well. So if training's something you're needing or you're thinking about, or perhaps could cost into some funding you're going for, please do consider reaching out. We can provide you information, and we've got discounts for academic startups and also multi attendees, as well as happy to create bespoke training as well. That's all for me. I'll hand back to Annalie.
Thank you, Neil. And next up, we have Sheena Fraser. She is she's the upstream bioprocess manager, also at the FlexBIO Scale-Up Center. Sheena, you're on mute. That's great.
Right, hi. Is my screen sharing yet?
Not just yet.
Not yet. I think I sent a request through rather than a stopping point. Give me a minute.
Okay, no problem. It's starting to share.
Oh, that's perfect. Thank you. Okay, so I think that's the starting point here is just to tie into what Neil has really just introduced us to, is the fact that the IBioIC's FlexBIO Scale-Up facility is here to support a broad and diverse range of bioeconomic projects and commercial development plans. The strategy here is really to offer a expertise across industries, across sectors, so that we do not necessarily selectively specialize in any particular aspect other than the umbrella of upstream processing or downstream processing. And the title itself, I think, conveys that from batteries, bioplastics, burgers, biomass, and building skills. It really is a broad base that we work from at FlexBIO. And the key hubs of scientific knowledge that we build or develop around shows the net zero focused or circular economy focus that Scotland itself has to develop technologies that are there to solve problems of the future. And that feeds into things like agri-tech or clean tech, food tech, and other waste valorization processes and projects. These technologies are developed as food or drink, pharmaceutical, sustainable chemicals, bio-based materials, and renewable sources of hydrocarbons as well. So from that point of view, it's not only around Scotland that we focus, it's beyond that. It's the broader UK. It's feeding into the technology hubs that are UK-wide as well as European-wide as well. And we've got a very small team at FlexBIO, but we've got a diverse set of expertise that comes through and translates into our knowledge exchange and project support that we offer the industry at large. And some of the examples, I think, to come into that, one of the key things that we do or have demonstrated at FlexBIO is how we change the waste, or what would traditionally have been considered waste industry-wide, into valuable projects and products. So the Whiskey, I think, project is a flagship example of that support and expertise. And this particular case study that I'm going to talk you through today is a collaboration between three startup companies that were Horizon Proteins, MyAlgae, and Biotechnologies that came together to try and find a more circular economy-based solution to using alternative waste streams that come out of industries like whiskey and other distillery efforts in Scotland and beyond. And here they looked at the distillation of pot ale, which is the second waste stream in the whiskey manufacturing process. The first would be the draff, or the grain that comes off processing and extracting sugars. And pot ale is the second waste stream that comes out of the distillation process or fermentation of that high sugar content pot ale. Most of the sugar has been converted to alcohol at this point, but there's a lot of nutritional value left in that pot ale. So how can you create valuable products out of that? And their IBioIC's role in this particular project was to work with Horizon Proteins, which was a Harriet Watt-based spin-out, and it was looking at the extraction of protein from draff using downstream processing methods. And the IBioIC assisted in scaling up that process using hundreds of liters of pot ale to develop a protein-based product that was then used to supplement and support aquaculture industry. The second example here that we collaborated on was to support MyAlgae. Now, many of you may have heard of MyAlgae. They've come to the fore in the industry as they have gained momentum in their scale-up of their commercial projects and processes, and in recognition for their efforts in net zero focus. So I'm going to just dig a little deeper into MyAlgae's aspect of that collaboration, and their goal of trying to deliver a high lipid content omega-3 algal biomass product for agriculture as a feedstock. Now, there are a lot of challenges around working with high lipid content biomass, none more than the upstream processes of maintaining those algal components or the biomass itself in culture. As they produce a lot of lipid, they tend to float and or flocculate at the top rather than being mixed. So the challenges around solving for product and process development from an upstream perspective, as well as ultimately the harvesting side of things. How do you harvest that algae and maintain the high lipid content without disrupting and breaking the cells? And how do you scale those different methods? So IBioIC's role and collaboration here has been long-term. We've been collaborating with them since 2015 to today, where we are still working with them to process biomass and improve the downstream collaboration or downstream clarification of their fermentation broth and processes. Now, MyAlgae has advanced its technology to the point that it has raised several rounds of investment, venture capital investment, and built a large-scale facility where it is commercializing this technology as we speak. Another key company or organization focused on biomass that IBioIC has supported and collaborated that may be familiar to many of you is Enough. That's a mycoprotein production company. They came to IBioIC back in 2015 when they were still named 3FBio. This is one of the companies that was incubated through the IBioIC, and IBioIC assisted in raising those initial rounds of investment that have supported the commercialization of their microprotein process. Now, what they were aiming to do here was to develop a zero waste process, looking at a dual stage production fermentation where you would produce microprotein in one stage and ethanol production in a second stage. So the IBioIC collaborated with 3FBio and Enough to develop these processes at small scale using our DASCIP systems, and also scale up to 30 liters, which was our largest capacity that we had access to at the time, as well as supporting them through knowledge exchange and commercial networking and support down the line. They use this proof of concept work that was originally done in collaboration with IBioIC to raise Series A, Series B, and various grant funding that has enabled them not only to build their own commercial plant in Glasgow, which has since been decommissioned, but also subsequently to partner with industry leaders like Cargill and build commercial scale facilities up to 150,000 square meters in the Netherlands, co-located with their feedstock supplier, which is Cargill. So this is the kind of support that industry gets from IBioIC and FlexBIO, not only the initial proof of principle through FlexBIO and the expertise from a technical perspective, but also the networking and engagement and business support from the IBioIC in general. Another example where we're looking at biomass here, an exciting project is cultivated meat. It's coming to the fore industry-wide. And here we worked with the Roslin Technologies, who are a Scottish-based company that are developing stem cells for cultivated meat. Now, their goal was to demonstrate that their pluripotent stem cells could be maintained in a pluripotent state as you scaled up their process. And ultimately, we have worked with them on a number of different cell types. They've moved from different species, porcine through to bovine, and other cell sources, as well as mesenchymal stem cells, as well as their pluripotent stem cells. So Roslin Technologies has recently raised their Series A funding, and Series B is I think underway at the moment to commercialize these cell lines globally. And our support is ongoing with them as we do test new cell types and cell lines in bioreactors going along. This is the foundation of our mammalian cell culture platform, and we are expanding on it to support the leather industry as well as other pharmaceutical grade products. To change completely from the biomass side of things, some of the other projects that we've been involved in are to demonstrate that you can find sustainable alternatives to the traditional acrylic products derived from the oil and gas industry. And this example that I have up on the screen at the moment through Manchester University was to develop a carbon capture method, also growing algae here, microalgae, and to convert the fossil-based, I suppose, to convert the media components that are available to this fungi into a perspex precursor. So these were engineered strains that were able to adapt and transform the media components into acrylic materials. So here, the expertise that we had is to use our Dasket platform in a design of experiments to improve the feeding strategies and to maintain the process and extend the process and optimize the carbon capture and transformation of those products and materials into plastic alternatives. Publications that came out of this are highlighted at the bottom of the screen. Feeding on from algae is not only the microalgae that we've been involved in, but we have been instrumental in introducing to the seaweed industry, which is a large industry in the UK as well as Scotland. It is how do we use our natural resources from the ocean and transform them into valuable products. One of the projects that we worked with was a collaboration with a company called Oceanum, who extract high purity nutraceuticals, sugars from sustainably sourced seaweed. The problem with seaweed is that it is very seasonal. The challenge is that in order to process it, you need to stabilize it so that you can maintain your supply chain throughout the year and extract the most value out of that seaweed. Here, stabilization methods have been used, traditional ensiling methods of treating the seaweed with high acid concentrations to actually prevent microbial growth. There's been a transition towards using lactic acid bacteria for a more natural, sustainable process where the acid produced by the lactic acid bacteria do a similar job to traditional ensiling. But these methods are known to degrade and denature the valuable compounds and nutraceuticals. So Oceanum were interested in finding an alternative method to this and approached FlexBIO to isolate novel microbes, to try and develop a novel process that was at a more neutral pH that could stabilize and sustain that seaweed so that you could then throw it into the extraction of more valuable metabolites going forward. And what we did there is through small scale studies, we isolated 2 bacteria that selectively metabolized a sugar that was not required, or they had no interest or value in it, which was mannitol, and used that to actually create an environment that stabilized the biomass in a neutral pH, eliminating and preventing growth there. This was the first introduction of solid state fermentation to the industry and the use of a solid state fermenter that we have since used and commissioned to support a number of different projects in the industry and to develop the know-how on solid state fermentation and its commercial implementation. And the last 2 examples I'm going to touch on are sort of more extreme waste scenarios. I'm just going to cover them at a very high level. I know that my colleague who is coming online afterwards from the Genomics Foundry Center is going to highlight these 2 case studies as well. But FlexBIO's role here is in a collaboration with the Faraday Institute and the Economics Genome Foundry, as well as University of Edinburgh, is looking at how to use a biorecovery method, a microbial method to extract and pull out valuable metals from battery waste. So this is part of the battery recycling and zero waste initiative from that perspective. So here we scaled a process that was developed around cell lines that Genomics Foundry, part of the collaboration, delivered and used that process to scale not only the upstream method to 30 liters, but also the downstream extraction of those heavy metals from the waste and fermentation process at large. So this is a project that has gone on to deliver a number of publication and presentations, as well as being awarded a prize externally as well. And on the same vein as that is using waste. Another example that I can highlight here is a project that is ongoing at the moment through a company called Sustainable Extraco, and its collaboration with the University of Edinburgh and Edinburgh Foundry is how you can take, I suppose, high polyethylene and polyethylene carbon composites, which are really difficult to recycle, how you can extract the materials and the components of that and produce high value, potentially food ingredients or therapeutic ingredients out of what is a existing waste stream, as well as transforming the hydrocarbon waste from industry, the oil and gas industry, into something that is more sustainable. And Sustainable Extraco have developed a novel method called presolysis, which depolymerizes these really difficult to degrade composites into monomers, and using a recombinant microbes, these monomers have been transformed and translated into the building blocks of high value food ingredient components, such as flavors, aromatic components, vanilla, etc. So this is the project that is ongoing at the moment, and I will leave the details of the strain development and other highlighting aspects of these last two case studies to my colleague who is up next.
Thank you, Sheena. I will share that at the end of the presentation as well, the QR code and how to join us. I am now going to hand over to Rennos Fragkoudis, who is the head of the Edinburgh Genome Foundry at the University of Edinburgh.
Thank you very much for the kind introduction. That was one of the best expressions of my name that I heard in the 30 years I'm in Scotland. So thank you for not making it wrong. Thanks for the organizers, both IBioIC and Copilot, for inviting us to talk a little bit about what we do in the Edinburgh Genome Foundry. So it's going to be a quite high level, and I would like to thank Sina for covering a lot of the information that because we work together with IBioIC and FlexBIO. So I didn't know who everybody was going to be here today. So initially, where are we? So this is a map of Edinburgh. This is a very well-known view of the Arthur Seat, and this is the Edinburgh Castle. This photo is a lie. We don't have sun like that very often in Scotland, okay? But we're based at the south of Edinburgh in the King's Buildings campus of the University of Edinburgh, and we are in this building here. We consist of a small team. We are a team of 5, but we have a lot of very cool robots that they assist us to deliver a high throughput. Let me get my laser pointer up. We are delivering high throughput innovation that accelerates the discovery process for a variety of different projects. What's a big advantage of our platform is that it's species agnostic, so we can work from anything from a microalgae all the way to porcine stem cells for artificial meat. Why biofoundries matter? This audience, I don't think, needs an introduction to bioeconomy, but in essence, we require a bioeconomy that's accessible to everyone, that's able to address grand challenges like securing the food we're eating, ensuring that our population is healthy and ages in a healthy manner, and even producing the energy that we are needing for society today in a greener way and protect the environment. And if in the process we can generate added value to the economy as well as green, what we call green jobs, this is obviously an added bonus. And what we do in the Foundry is we play with the currency of bioeconomy, if you like, which is DNA. And this is the very well-known cycle of engineering biology, going through the design, build, test, and learn phases. And this is what the Foundry is doing. We're assisting people through all the phases of this cycle, and we're trying to accelerate it as much as possible with the help of our knowledge and the robotic systems we have here at the university. So this is a video showing, it's not a drone, somebody asked me before, it's just a 360 camera above our platform. And you can see it's a quite massive platform, and we have a lot of plans for expanding its capabilities. But in essence, we're using robotic arms, liquid handlers, as well as automated incubators and instruments for phenotyping to allow us to deliver our services to the customer. And our aim, and our customers come from a variety of backgrounds, so they can be academics, both internal and external, as well as people from companies. And same as FlexBIO, we're also trying to break even, so we're not a for-profit organization. So our aim there is to support people accelerate their discovery process. So I'm going to briefly touch on how we are assembling multiple constructs in a high throughput way. If we're operating in an automated way, fully automated way, we can produce more than 2,000 constructs a week. And we're coming in the step before the scale-up facility because we allow our academic and non-academic partners to test and sift through, if you like, through a large number of different varieties of a construct or of a bacterium or of a mammalian cell, and pick the one that has the optimal properties and then take that to the scale up. So for us, everything starts and finishes with our software. This is something we're very proud about, and it's one of our USPs. We have software for the design of constructs, for the simulation of constructs, the software that allows to communicate with the robots, as well as software that allows us to deliver our research innovation in a responsible manner. So once all the design process involving the software has been completed, the robots take over, and we do not use pipettes, but we use acoustic or liquid handlers, and we do not use that many people, but we use robotic arms. And once this is complete, again, the software is helping us to do the QC of our product, which is the DNA. On top of that, we're able to develop a lot of bespoke assays, anything from fluorescent luminescent assays to real-time PCR in the near future digital PCR, as well as automated microfermentation. And all this capability allows to deliver a variety of projects, and Sina previously touched on a couple of the projects that we've done on lithium-ion batteries and plastic upcycling. So on this first project, we came before the FlexBIO facility and worked with Louise Horsfall's group here at the University of Edinburgh and used this instrument called the BioLector, which consists of a number of mini fermenters. So each one of these chambers that is here is a mini fermenter, and you see two opaque dots. These are called optodes, and they're measuring dissolved oxygen and pH. And this, in essence, allows to very nicely go through a large number of different strains in this case to find out how using different leachate types with different contents of metals, it's going to affect their growth rate, their requirements for growth in terms of pH and feedstock and things like that. And from that, Virginia and Luis, they picked the more promising strains, which were then scaled up. You also heard about the study from trying to upcycle plastic from sailing boats. So this is a project we've been working for quite some time with Joe Sadler group here at the university. So Joe had a very high-profile paper which showed that they could use plastic to produce vanillin. So at the moment, we are taking it a step further and we're working on a project that allows us to see if we can further improve the pathway. And I'm not going to stay long on that because we already covered it. I'm going to move to something very unique. This is the only instrument of its kind in a European university. It's called the Beacon. It's used exclusively with mammalian cells in the Foundry. And it's groundbreaking technology. The way it works, this chip that you see on the bottom right here, the glass panel, it's covered with semiconductor material. So when light hits it, it forms a localized electrical field that moves cells within these chambers called nanopens. And not only that, but when we are done at the end of the of the process, we are able to again use light to take these cells out of their chambers, put them in a 96 plate, and then scale them up. And at the moment, in the Foundry, we can scale up to 125 ml shake flasks. So then we need our colleagues in FlexBIO to take them to the next level, 2 liters. We use that for a variety of different projects. One of the main applications when it comes to industry is developing novel cell lines that allow biologic production. So more than 20% actually of all new medicines are biologics. And last year, the market was valued at more than $500 billion. The prediction is by the end of, by 2030, this will exceed $1 trillion. And you can see the problem here on this video that just played for you. This was supposed to be a very homogeneous population of CHO cells. As you can see here, this could not be further from the truth. So using the beacon, we can compress the timeline. The process of developing a novel cell line takes 2 to 3 months. We can do it in 5 days. And we can accelerate the discovery process by picking the clone with the ideal properties much, much faster. So as you can see on this panel here on the right-hand side, this graph highlights how cells are growing. And you can see that all the cells, they seem to be growing exactly the same way. But when it comes to producing what we want them to produce, which in this case is IgG antibody, you can see that this could not be further from the truth. So we can pick the clone much faster. Also, most recently, we received funding from BBSRC to develop some novel therapeutics. So to do that, we need to be able to check, to be able to control if a gene is expressed to the correct level, at the correct tissue, and at the correct time. And this is one of the ways that allows to control when a gene is expressed. So together with Susan Rosser's group and Adam Moll, a postdoc in that group, we developed this system which utilizes the power of aptazymes. Aptazymes are riboswitches that when active, they don't allow a gene to be expressed. But through the binding of a small molecule ligand, when they become inactive, they allow the gene expression to proceed. And this is, I want to reiterate that this is a proof of concept study. We wouldn't imagine in the society where everybody is living on caffeine, a switch that would work based on caffeine. This is what construct looks like. And then we put it in cells and we try the cells on the beacon. So the M-cherry here highlights that the cells have the construct that we have created. And on the right-hand side, we're floating caffeine through the system. You can see that the cells are switching on. So expression is taking place. And when the caffeine is removed from the system, then this is switching off and going back to normal. So before I hand over to the next speaker, I wanted to make you aware of the latest workflow that we incorporated on our beacon. So we have three workflows. You've seen cell line development. The other one is antibody discovery, when you can discover hits within a day. And the last one is development of CAR T cells. And in this instrument, we can, within a number of days, put CAR T cells, assess their ability to secrete cytokines, do proliferation assays, but also check if they can kill tumor cells. And here you can see one such example. So the green cell here is a tumor cell, and as the CAR T is attacking it, you can see it turning red. This shows that the caspase-3 is activated, and therefore what you're witnessing on your screens is the killing of a tumor cell by a CAR T. Most recently, and as part of the hub, we received strategic funding for the Foundry that will allow us to expand our capability for automated mammalian cell culture. So the aim is to take more hits from the beacon, scale them up to the 125 ml shake flask, and then we can hand them over to our colleagues in FlexBIO or other scale-up facilities around Europe. So this is expected to be on the ground in about 11 to 12 months, so around this time next year. And obviously, I lied when I told you there was only one sunny day. There were two, and this is the second one. And I would like to thank you for your attention. And if there are any questions, I would be happy to answer. Please get in touch.
Thank you very much, Rennos. And the last speaker is Mark Bustard, who is the CEO of IBioIC.
Thanks, Annalie. Share my screen, hopefully everybody can see that. So a little bit different topic just to finish up on, which is really an opportunity to collaborate and also to raise the profile of a project that we're involved in, a European project called the European Circular Innovation Valley Project. So this is a typical EU funded project. It's got 18 partners. And you can see some of the regions that are involved there. Some very strong bioeconomy regions in there, and also some regions that are obviously still developing their bioeconomy strategies moving forward. So it's been very interesting working alongside them. For Scotland's representation in this, we have Scottish Enterprise, which is the development agency for Scotland, and also IBioIC is the innovation centre. So just to raise a little bit of awareness. I won't dwell on this. I think we all understand circular economy and what we're trying to achieve, particularly from a bio-based focus. From Scotland's perspective, our main interests in this project come from our bio-based and our industrial biotechnology cluster, which has been in significant growth over the last few years, and how we reach out and collaborate with others. The critical bit is obviously it's great for us to be involved in European projects again after a period of disappointing absence. And so that whole collaboration and exposure to other networks and innovators and what other regions are doing is incredibly important for us. Just to a little bit of granularity on it. So these are the missions and the submissions. There will be funding coming. I'll tell you a little bit about that. But it may not be any surprise that the topics that you see on the screen have been identified by the 18 partners as priorities for them. Clearly, there are some clusters who have got particular strengths in things such as electrical or electronic equipment, steel, metals, mining. That's not for Scotland, obviously, but we do have an interest particularly in food and agri-systems, textile and fashion, the wider bioeconomy, and also water and wastewater. So we're involved in a number of these sub-themes, and we'll be looking to form consortia as the calls come through. We've just been 1 year or so into the project, so a lot of formation and a lot of strategy in identifying the areas for support. What will happen now is there will be 2 open calls at late TRL, so 6 to 8 approximately, and also 1 early stage call for lesser money as well. I think the critical bit is there will be over 16 million euros available to fund these collaborative research and development projects. And for those clusters who have their development agencies involved, they will also contribute another 7 million euros on top of that as well. So again, a good scale of activity. The critical bit here is we need a project to have at least 3 partners of the participating regions, but also those of you who are outside of those regions can also participate in this as well, but usually that funding will come from your local development agency as well. The scale of what we're offering at the moment is projects of up to 2 million euros, and the maximum that any particular company can draw down is about €500,000. The timeline, so obviously from it kicked off in September 24, a lot of work has gone in across the partners in working to identify these sub-themes, look at innovation strategies for the clusters and so on. The important bit to flag now is that in December, the first open call will come out for innovation projects, which are the sort of up to £2 million projects. And then for the experimentation projects, which are the smaller projects as well, of sort of a couple of €100,000. The call will then close in February, and then we'll work on developing the next set of calls. The call theme for the first call is textiles, wool, and fibrous materials. And so most of the clusters have an interest in that, and probably your region does as well. The main platform that we're using to help connect people up, other than our own ability to connect people, is the the B2Match platform, which you can sign up to using the QR code here and follow us in terms of what's happening on the project. It has its own dedicated website, and you're very welcome to join that and hopefully learn a little bit about the partners, about their interests and opportunities to work collaboratively to secure some of that funding. So that was really all I wanted to say today, just to raise the profile, and thanks for your interest.
That's great. Thank you very much, Mark. And also thank you to all of our speakers today and everyone coming along. Just to quickly wrap up, I just wanted to share a QR code with you to be able to join IBioIC. Along with all the benefits that Neil already mentioned, when you are a member of IBioIC, you can also attend free events organized by the Scottish Fermentation Network. And to mention that we also have a jobs board, so companies who are members can share their new opportunities, and we can advertise it on their behalf, and then students can easily find what those opportunities are. And lastly, I would also like to flag our annual conference, which will be next year between the 10th and 11th of March in Glasgow. So I hope that we will see you there. So thank you very much, everyone, and enjoy the rest of your afternoon. Bye-bye.